So good morning, everybody. We're gonna continue looking at the nervous system today. So let's just get into the warm-up, since the warm-up is kind of a review of the nervous system. All right, share screen here. All righty. So like I was mentioning on Thursday, we kind of laid out a kind of roadmap to the whole nervous system, and it's important to kind of be able to stay oriented as we go through the different parts over the next few weeks. So this first question was kind of, all right, which parts of these things here are part of the motor division? And there's still confusion. If you're one of the ones who still has some confusion here, I highly recommend kind of going back to that kind of roadmap and getting more comfortable with it. If it's so motor division, that's part of peripheral, which means it's not going to be the spinal cord or the brain. Spinal cord and brain are only the central nervous system, the CNS. Motor division is part of the PNS, peripheral. So then we knew there's the somatic motor, which was the one that does your voluntary muscles. There was the autonomic motor, which does your glands and involuntary, like smooth muscles. And then the autonomic was further subdivided into the sympathetic and the parasympathetic, which were subdivisions of motor as well. Efferent was a synonym for motor. I don't know if I mentioned this, the mnemonic people usually use when they're trying to learn this, it's like, you have the CNS here, you have the sensory, kind of the information going in, the CNS is the integration, and then you have the signals coming out, the motor. So this is also afferent. That's kind of the A signals are the ones that are arriving at the CNS. Motor is efferent, like that E. These are the signals that are exiting from the CNS. So that's the way people usually try to help themselves remember. Kind of afferent arrives and the efferent exits. So if that helps, you can use that. All right, let's get back to the warm-up. Okie dokie. Yeah, so afferent is not correct. Afferent and sensory are afferent divisions. This one, a lot of people also, so adrenergic, you should know this adjective. This is describing anything that is relating to adrenaline, which is also known as epinephrine. And then the related neurotransmitter is norepinephrine, also called noradrenaline. Anything else is not adrenergic. GABA, dopamine, serotonin, acetylcholine, glutamate, they have their own adjectives, GABAergic or dopaminergic or serotonergic or cholinergic or glutaminergic. Adrenergic is referring specifically to epinephrine and norepinephrine, also known as adrenaline and noradrenaline. If this question instead says cholinergic receptors combined, what would the answer be?
Acetylcholine.
Yeah, it would just be acetylcholine. All right, what is a node of Ranvier and what is its function here? Most people got this, although it was maybe a little unclear for some people. The node of Ranvier is like a little gap in between the myelin sheaths. And it's where you have like the voltage gated channels that can regenerate and keep the signal going, keep the action potential going. They usually happen about every millimeter along the length of the axon. Fastest transmission here, people did well. Again, it's going to be the ones that are the fattest and are myelinated. They're, we have a classification system. They would be like type A axons are the ones that are like the ones that are taking messages from your skin senses up to your brain and sending messages out to your skeletal muscles. Those are all these kind of type A fibers that are big, large diameter myelinated that can send action potentials like 150 meters per second, as opposed to like your like class C fibers, which are small diameter unmyelinated, where the action potential is kind of poking along at like a meter a second or something. So, all right, this one people got. CNS, brain and spinal cord. Woo, 100%. All right, so speaking of brain and spinal cord, actually speaking of all of this, before we really get into the brain and spinal cord, which is where we're gonna kind of start our focus, we do need to talk a little bit more just about nervous system in general and the basic nervous tissue and the kinds of cells and some of the terminology that we're going to be using. So let's do that. So obviously, the kind of famous cells are like the neurons. These are also called like nerve cells, also called nerve fibers. Fiber because they often have long processes that come off of them. It's like, fiber, a long, skinny thing. So nerve fibers, nerve cells, neurons, they're all just, these are kind of the most famous of the cells that make up your brain and spinal cord and peripheral nervous system, but there are other cells as well. They're also what are called neuroglia. Glia means glue. These are like the cells that are kind of filling the space, supporting the neurons, doing a variety of things. We're going to talk about different jobs they do in just a second. Sometimes they're called like supporting cells of the nervous system, but neuroglia or glial cells is the more common term for them. These are smaller and more numerous than neurons. For instance, in your brain, half of the volume of your brain is neurons, but the neurons only make up maybe like 1/10 of the cells, because there's tons of these neuroglia cells doing all this metabolic support and myelination and all this other stuff. So let's just briefly, I don't want to spend too much time here because we have lots of ground to cover, but I think it is worth having a kind of introduction to them. You've probably met them already in anatomy. The supporting cells that you find are different depending if you're looking in the CNS or the PNS. So we're gonna, in fact, let me just go a new page so I have more room. So one of the jobs that these glial cells do is make the myelin sheaths. The ones in the CNS that make the myelin sheaths are called oligodendrocytes. In the PNS, you also have cells that make myelin sheaths, but they're got a different name and it's in a different anatomy and some different roles as well. So I'll talk about in a second. They're called Schwann cells. Schwann cells, I'll briefly talk about. So oligodendrocytes kind of reach out. They wrap their membrane actually kind of like a jelly roll around the axon to kind of create an insulation. I'll kind of show you a picture of it in a few moments. Schwann cells are a little different in that the entire cell kind of becomes one myelin sheath. Like if here's an axon, the whole it's a cell that wraps around and becomes, again, kind of like a jelly roll around the axon. There's going to be a little node of Ranvier where there's a little gap in between. One of the things that Schwann cells can do that's in addition to making myelin sheaths is they can actually act as an insulator. I talked about how there are unmyelinated axons, right? An unmyelinated axon is still, though, you don't want to have it touching another axon because it's going to short circuit. It would be like two wires touching each other. So you can also have these Schwann cells playing a role as just insulators. So you have like some axons running through the myelin sheath, or the myelin, it's not making a myelin sheath, but it's separating the unmyelinated fibers from touching so they don't short circuit. So that's something a Schwann cell can do as well. And then the other thing a Schwann cell can do is it can help if there's been damage to the neuron. So for instance, let me draw this. Let's say here's a neuron with an axon. And you have these myelin sheaths that are made out of these Schwann cells. And injuries happen. So bam, you just got cut and it happened to slice through an axon of a neuron that was going off. Maybe it's taking messages in from your skin. And now things feel kind of numb where it used to innervate because the messages aren't getting to you. When there's damage like this, the neuron, the cell body, and the axon that's proximal is going to be okay. But downstream, distal to the injury, the axon is going to die. There's going to be phagocytes that come in and kind of clean things up. But one of the things that happens is these Schwann cells, basically, they can create like a tunnel, a guide. And that guide then allows this axon to regrow and hopefully find its original target and connect up and be functional the way it was before the injury. The term they use is Wallerian degeneration. This idea that the axon that's farther away from the injury kind of degenerates and gets cleaned up, but then the Schwann cells reconfigure as this guide that allows the neuron to regrow and hopefully connect up. Depends obviously how clean the injury is. And these things grow pretty slow, but it's, if I'm probably lots of you have experienced it. I've had injuries where things have been numb for months and months and months, and all of a sudden it's like, holy crap, I can feel it again. Because eventually the axon made it all the way and reconnected to the target, and now things are back to where they were, or at least close. So that's something that Schwann cells can do as well. They can assist with the repair after injury. So one thing glial cells do is they make myelin. And then we saw again in the PNS, they also help insulate unmyelinated axons from touching each other, as well as assist with repair after injury. In the PNS, the only other glial cell that exist are satellite cells, which I'm not going to talk much about because we don't really know a whole lot about. Satellite cells seem to provide cushioning and metabolic support for the neurons in the peripheral nervous system, but we're not going to go any deeper into that, so don't worry about them. On the CNS side,
really quick, can you repeat that? They cushioning in what?
like metabolic support, kind of cleaning stuff up, providing nutrients, keeping the solutes floating around them, appropriately balanced. So in the CNS, we're going to have a variety of more interesting glial cells. There's going to be astrocytes. Astrocytes, they get their name because they have little processes that come off, so they look like little stars. Yeah, an asterisk gets the name asterisk. Astronomy, astro means star. Astrocytes are cells that look like little stars. These do a lot of interesting stuff. These, help maintain like environment. Yeah, you spell that right. Environmental milieu. I think that's how you spell it. No, it's IE, like that. Dagnabbit. Kind of the environment. We talked about how when you have a synaptic event, one of the ways synapses are cleared is diffusion out of the synapse. It's like, where is that neurotransmitter going? It's going out there. If it gets into some other synapse, it's going to cause problems because it can bind to receptors. So they're kind of cleaning things up. They're like kind of custodians that are cleaning up leaked neurotransmitter that shouldn't be out there anymore. They are adjusting the levels of ions, because obviously we saw that the concentration of ions out in the extracellular fluid is critical in determining the membrane voltages. So that's part of what the astrocytes do. Metabolic support to the neurons. They actually transfer the sugars to the neurons to give them energy. Help create the blood-brain barrier. So like if you see blood vessels in the central nervous system, they have all these little astrocytes reaching down and creating this special kind of boundary around the vessels. So we should talk, let me talk a little more about the blood-brain barrier since we're here. So the basic idea here is there are lots of things that might be considered neurotransmitters if they're in your brain. Remember, we talked about adenosine. Adenosine might just be a raw material to make ATP, or it's a neurotransmitter. So you can't just have random molecules floating around the neurons in your brain because they might actually be doing stuff. So there is this really tightly regulated boundary between your blood vessels and the tissue spaces around your brain. And only very specific things can move out of the bloodstream into the space around your neurons. So most capillaries are very leaky. Like the whole point of capillaries is where materials can enter and leave the blood. So most capillaries allow things to go out, to come in. That's the whole point of capillaries. In the brain, it's much more tightly regulated. So very tightly regulated. Only very specific things are allowed to move out of the bloodstream. Obviously, sugar can go out. Things like oxygen can just diffuse. But one of the problems when you're designing drugs that are supposed to be effective for the central nervous system is even if you found the perfect drug that would do a really important job, if it was, delivered to your brain, the chances are if you give it to somebody or inject it into them, it will never get to their brain because things don't move out of the bloodstream into the space around the neurons. Like when people are designing CNS therapeutic drugs, they have to worry about two things. One is this going to be an effective drug? But second, is it ever going to get to the brain in the first place? Because you only have very specific kinds of molecules that are going to be allowed to leave the blood and get into the brain. So, and this blood-brain barrier is, at least in part, created and mediated by the astrocytes. So, blood-brain barrier. We'll see the other place we're going to see a very tight boundary between the blood and the tissue is in the testes. When we get into the male reproductive system, there's going to be a blood testis barrier actually as well. That will be important when we get there, like at the very end of the semester. All right, so CNS astrocytes, helping maintain that environmental milieu, or the external environment is probably an easier way. I like using these French words because they're fun to say. Milieu, Pluton. I don't know. It's prettier than like some other languages. Like, yeah. The metabolic support for neurons, like I said, critical for like just delivering, sugars and stuff to the neurons. And again, blood-brain barrier. They seem to be doing other things as well. They actually can communicate between themselves. They have slower electrical signals that they use to talk to each other. So even though the neurons are the rock stars of your nervous system, these astrocytes seem to actually be communicating with each other, adjusting levels of like neurotransmitters floating around to actually change the operating point of things. So they're not purely like custodians. They seem to be a little more active, but we're going to focus on the more exciting role of the neurons when we look at processing and things like that. All right, so CNS glial cells. There's more. I've ran out of room here, so I'm going to go on to a new page. Whoops. So glial cells of CNS. Oligodendrocytes. Again, making the myelin. Astrocytes. Doing all that stuff I just said. Another important one are going to be the ependymal cells. So for these to make sense, you have to know that the CNS has hollow spaces filled with fluid. Again, if you've taken anatomy, you know that. Your brain is got all these ventricles, these big hollow spaces with a CSF in there. Your spinal cord has a tunnel going down the middle filled with CSF. And all those hollow spaces in the brain and spinal cord are lined with these little ependymal cells. They actually have little cilia that can help circulate the cerebrospinal fluid. They also play a role in creating and maintaining the composition of the cerebrospinal fluid. And we'll talk about that in a few moments. We'll talk about the choroid plexuses and stuff that make cerebrospinal fluid. They're going to be an integral part of those choroid plexuses. So ependymal cells lines hollow spaces of the brain and spinal cord. Finally, the microglia. These are basically phagocytes. Your CNS is what people call an immunologically privileged area. Like we're going to talk about the immune system, which in general functions everywhere in your body, but it doesn't function in the same way inside your brain and spinal cord. You know, part of the thing is like, you know, classic immune responses like inflammation would be a disaster. If your brain is inflamed, there's no place to swell and so you have these specialized little cells that can go in and eat things, clean things up, help remodel things in the brain and spinal cord. They're called microglia. People are looking at them more and more in depth now as we learn more about them. They're getting more and more interesting in how they actually come into being and the roles they play. So you might see them mentioned in, you know, scientific papers that you read that you're interested in about different things going on in the brain. You're like, what are microglia? Oh yeah, those were those little phagocytes, those things that are kind of the representatives of the immune system, as well as kind of cleaning stuff up in the brain and spinal cord. Now, beyond that, you don't need to know much about them, but you should know you should know, at least have a sense of who they are, what they're doing, because like I said, they are becoming more and more a focus as people realize that they're more and more interesting. So these are all the glial cells. You know, oligodendrocytes, we're not going to talk much more about, other than you should know that things are myelinated. Axons are myelinated to speed up the transmission. Astrocytes, we're not going to talk much more about, but you should know that they're there to maintain the external environment around the neurons, and that they're also providing metabolic support. And you should definitely understand the idea of the blood-brain barrier that they're helping create. Ependymal cells, we're going to actually see in a few moments as we get into the brain in a little more detail and look at the ventricles and aqueducts and central canal of the spinal cord and all that. Microglia, we're not going to talk much more about, but like I said, you should be aware that they're there and their basic role. All right. So those are the cells of the nervous tissue. A couple of more terms we need before we actually just dig into looking at the brain. Again, usually we have different terms, whether we're talking at the CNS or the PNS. So if I have a bunch of axons running together, so group of axons, right? And I could have a bunch of neurons and then they have their axons that are all kind of going along. So I am talking about this kind of bundle of axons, you know, and it's usually lots of different neurons that are all feeding in. So this thing here, it's like a cable of axons that are sending a message from one place to another. If I have a group of axons in the CNS, I call them tracts, axon tracts. I will elaborate on these in a little bit more because sometimes there's going to be tracts that just stay within a hemisphere, like association fibers. There's going to be commissures that go back and forth between the right and the left hemispheres of the brain. There's going to be projection tracts that are going between upper and lower regions of the nervous system. So there's different subtypes of tracts, but they're still called tracts. In the peripheral nervous system, they're called nerves. So this word, you know, has different meanings in different contexts. You know, I got a case of the nerves, you know, that's something different. This is just a bunch of axons. If you're looking at the sciatic nerve, you know, people have a sciatic nerve. It's carrying messages from your central nervous system down the back of your leg to operate the muscles in your leg, as well as bringing up sensory information. It's just a bunch of axons carrying motor and sensory signals. Your phrenic nerve is sending messages down to operate your diaphragm for your lungs. Your radial nerve is operating stuff in your arm. Your optic nerve is carrying messages from your eyeballs to your brain. So nerve is just a bundle of axons carrying a message from one place to another place. You know, outside of the CNS, obviously, so in the PNS. The other term, groups of cell bodies, groups of neuron cell bodies. If I'm in the CNS, there's this word they call the nucleus. Singular would be nucleus. You know, we're going to talk about the basal nuclei. We're going to talk about the amygdaloid nucleus. We'll talk about, there's an olivary nucleus. There's all sorts of nuclei. They're basically little clusters of neuron cell bodies in the brain, in the spine, in the brain mainly. So we're going to see nuclei, the lentiform nucleus. In the PNS, when you have clusters of cell bodies, they're called ganglia. Singular is a ganglion. Right, so, you know, the neurons that take the messages from your senses into your central nervous system, the cell bodies for those are actually outside the spinal cord. They're sitting near the spinal cord, but they're not in the spinal cord. Those are going to be like the dorsal root ganglia. Whenever you see that word ganglia, it just means cell bodies that are not in the brain or spinal cord. You know, again, if we were in anatomy, we'd put a lot more energy into learning about the different ganglia and where they are and blah, blah, blah. But for this class, you should just know what the term means, really. We will be looking at specific nuclei in the brain and what roles they play. A little more, let's focus now more like on the CNS and some terms that are going to be coming up. We'll look a little more at these tracts, the white matter, and we'll also talk about some other terms we use to describe where you have cell bodies. Yeah, so let's say this is a person, the brain inside their skull has these kind of two hemispheres. That's what this is kind of, this is representing kind of like the two hemispheres of the brain. Right? Everybody's probably got this sense of the brain as this big oversized walnut with these two halves, these two hemispheres, the cerebral hemispheres. We'll look at the cerebral hemispheres in more detail in a few moments. When we think about where the neuron cell bodies are, there's going to be deeper nuclei. So maybe I'll draw it right here. Kind of like that. So there'll be some areas in the brain where we have nuclei, clusters of neuron cell bodies that are little islands deep within there. But then there's also going to be what we call the cortex. So this is, and we're going to call this gray matter. Gray because it looks kind of gray as opposed to the myelinated axons, which look white because myelin is kind of fatty and white looking. So cortex, thin surface layer. So the cortex is maybe 5 to 7 cells thick, making this layer right on the surface. Like if you are holding a brain in your hand and poking at it, you're touching the cortex. Cortex means bark, you know, like the bark on a tree. So when we think about the gray matter, which is mainly the neuron cell bodies, it can't, there are a few cases where gray matter is actually referring to unmyelinated axons, but let's not worry about that right now. Gray matter is basically going to be the neuron cell bodies. It can either be deeper nuclei, like pockets of neuron cell bodies and little islands deeper within the hemisphere, or on the surface, creating this kind of wrinkled sheet. Yeah, the reality is the cortex has to like get shoved into your skull. So it gets all wrinkled up as you shove it up into the narrow confines of the inside of your cranium. But if you flattened it out, it would be like kind of a tablecloth or something. This is where kind of the calculations are happening. This is, you know, where you have the axon hillocks, you know, where all of the IPSPs and EPSPs are being integrated and trying to decide whether or not it's time to send a message down the axon to some other place in the neuronal circuit. So the axons are called white matter because they're myelinated, and axons that are myelinated look white. So the other thing we're going to talk about is white matter. This is just going to be myelinated axons. I mentioned that there are some different categories of tracts, axonal tracts. So let me just put those out here now. We can have association tracts. fibers. They're basically just interconnecting stuff within a hemisphere. So some of the axons are interconnecting things staying within a hemisphere. There are commissures. That are connecting the right and the left sides. And then the projection tracts are ones that are going between higher and lower brain regions. So you can kind of get this idea. These things I've drawn in green, red, and blue are axons, myelinated axons. The green, the association tracts with staying within a hemisphere. The commissures interconnecting between the two hemispheres. I should mention of the commissures, the main commissure is, if you've been in anatomy, you know what it is. What is it called?
The corpus callosum?
Exactly, the corpus callosum. Now, you should know that the corpus callosum is the main commissure that interconnects the right and left hemispheres of your cerebrum here. There are other ones. There's an anterior commissure. There are more than one. There are other commissures that are not as big and bulky, but the corpus callosum is the major band of axons that connects between the right and left sides of the brain. Okay. So now, any questions up to here? Okay, so let me share. All right, so, again, when you think of a brain, you think of this big, wrinkled up, oversized walnut. And when you look at it like this, it's really hard to get a sense of what are the different sections and how do they relate to each other, and what's higher, what's further up in the brain, what's further down in the brain. It's like all looks like a big mess. And it's because this whole thing has to get all wrinkled up and shoved up into your skull. What we're going to do is look at the development of the brain. Basically starting from this little hollow tube. So like, here, like a month into development after you've been conceived, what's going to become your brain and spinal cord is just this little tube with some little swellings. So let's take a look at how this works. So the basic idea is like a month into development, you're not much more than a little frisbee of cells, with those three germ layers, the endoderm, mesoderm, ectoderm, that are starting to differentiate into different parts. Again, this should all sound familiar from Bio 110. The ectoderm folds, so you've got like ecto, meso, endoderm. Remember the three germ layers. The endoderm is going to fold around and become the lining of your gut and stuff. The ectoderm folds in to become this hollow tube at the top. So this is happening maybe about a month after you, right? First, you were a twinkle in your parents' eye, and then an egg and a sperm got together, and then the fertilized egg starts going through mitosis once it's become a zygote. And ultimately, things are starting to become things that are going to actually be you ultimately. So this dorsal hollow nerve tube starts getting little swellings. So now, instead of looking at it from the side, I'm kind of looking at the top of the Frisbee. This is that dorsal hollow nerve tube here. And this is what's going to become your brain and spinal cord. This part here is just going to become the spinal cord. These part here, this is going to be the forebrain, midbrain, and hindbrain, or the prosencephalon, mesencephalon, rhombencephalon if you're in anatomy. These are what are going to become the different parts of your brain more proper. It's important to remember that it's a hollow nerve tube. So as all this is happening, we're going to maintain this hollow space inside, lined with ependymal cells, filled with cerebrospinal fluid. That's going to be important for our story of the brain. So this is where we're going to start. Now I'm going to take this embryonic hollow nerve tube and we're going to follow the fore, mid, and hindbrain and see what they become. And again, I think this is a useful exercise because it helps you keep track of what are these different parts and how do they relate to each other. So the forebrain, and this midbrain, hindbrain, and again, the other parts just become like the spinal cord. And like I said, in anatomy, it's the prosencephalon, mesencephalon, rhombencephalon, but it doesn't matter for this class. So the forebrain is going to become the cerebral hemispheres as well as what's called the diencephalon. So let's follow that. So those cerebral hemispheres that I was just talking about a few moments ago with the cortex and the nuclei, that's coming from the forebrain. So this is going to become my cerebral hemispheres. Or my cerebrum. As it's growing, these get really enlarged because those cortical surfaces are really have a lot of surface area. So they start getting all kind of wrinkled up as well as they start growing, as they try to fit into your skull. So cerebral hemispheres. The forebrain also becomes what's called the diencephalon. The diencephalon is going to give rise to the thalamus, or thalami, I should say. There's two of them. Hypothalamus, epithalamus. We'll look at those in more detail later. But the diencephalon, the thalamus, is like kind of considered gateway to the cerebrum. It makes more sense now that you can see that, it's there at the gateway. To get up into the cerebral hemispheres, you have to go up through the diencephalon. The midbrain is just going to become the midbrain. We are going to talk more about stuff you find in the midbrain. There's the colliculi, the substantia nigra, but we'll look at that in more detail a little later. So midbrain, and then the hindbrain is going to become two things. It's going to become the pons, the medulla oblongata, and then it also gives rise to this thing that kind of hangs off the back. In fact, let's do these first. If we look at the midbrain, the pons, and the medulla, these together are going to be called the brainstem. You know, you can kind of think about it kind of like almost like the stalk of a mushroom. And then the thalamus is kind of the top of that stalk there. And then the cerebral hemispheres kind of arch out kind of like the cap of a mushroom. So that is this basic idea here. The hindbrain also gives rise to this thing called the cerebellum, which hangs off the back of the brainstem. So this picture that I've drawn here is kind of right and left. Everything here would be like if you're looking at a person that's kind of looking at you, the right and left hemispheres here. But if we look at the brain from the side, so a side view of the brain, then there's this other thing that derives from the hindbrain called the cerebellum, kind of hanging off the back of the brainstem. We will talk about the cerebellum in more detail later. It's basically about this kind of smooth coordinated movement. Important to not confuse the words cerebrum and cerebellum. They look kind of similar. Cerebrums, these are the hemispheres, the two main hemispheres that are the kind of uppermost parts of your brain. Cerebellum is this doohickey hanging off the back of your brainstem, more involved with motor stuff. So don't get those words confused. Okay. So I've been mentioning here are all these different sections. I was also clear that this hollow nerve tube, that we don't want to forget that it's hollow. So as all of this crazy folding is going on, this hollow space stays hollow, although it gets much more elaborately kind of stretched out. So let's take a look. Maybe I'll use blue. I mean, again, in anatomy, you actually get into the, in fact, let me draw this on a new page. It'll make it prettier. Too high. And my cerebral hemispheres. My diencephalon, my midbrain, my pons and medulla, going down to the spinal cord. So this is that same picture, kind of, sort of. Then the hollow spaces. Again, if you've been in anatomy, you know the lateral ventricles and the foramen of Monroe and the third ventricle in between the thalami and the aqueduct of Sylvius and the fourth ventricle and the central canal of the spinal cord. You know, for our class, you do not need to know the specific names of the different ventricles and aqueducts, but you should know that we have this system of hollow spaces that are within there. So we call ventricles and aqueducts, etc. They are all filled with a fluid called cerebrospinal fluid. So what we're going to do now is we're going to talk about CSF, how it's made, how it flows. We're also going to talk about the different membranes, the meninges that wrap and hold the brain as well within the skull. Can also, all right, so let's just start with the CSF. So CSF is produced within the ventricles of the brain. Ventricles just mean these hollow spaces. And it's produced at these structures called choroid plexuses. So a choroid plexus it's basically leaky capillaries. You've got capillaries that are then also, though, covered with these ependymal cells that are going to kind of really adjust what is coming out. And the basic idea is you have leaky capillaries, which means, you know, plasma is going to like leak out. Although the exact composition will be adjusted by the ependymal cells because they're ultimately maintaining that boundary between these capillaries and the space inside the ventricle. So CSF is a lot like plasma because it's fundamentally leaked plasma, continually produced. right? Because it's just made by constant leaking from these leaky capillaries. And again, the actual composition is being adjusted by these ependymal cells. So if we go back to this other picture now, maybe I'll make them red. They're here in the floor of the lateral ventricle and the roof of the third ventricle, up here in the fourth ventricle. Yeah, etc. So these little things that I've drawn as little red squiggles here, those are representing the choroid plexuses within the brain that are constantly making CSF. All of these hollow spaces are lined with ependymal cells. So those little cilia on the ependymal cells can help circulate and keep the CSF kind of flowing around in there. But obviously, it's got to go someplace, because if it's constantly being produced, if it was in a closed place, then your brain would inflate and you'd be in trouble. We'll look at some pictures of that in a little later, hydrocephaly actually, where the CSF drainage is blocked and the brain actually does inflate like a big balloon. It's kind of horrifying pictures of little babies that get born with their heads all inflated. So what we're going to do now is continue with this picture. So far we've just talked about the inside of the brain. Now we're going to go to the outside of the brain and watch where the CSF is going to flow to. So the outside of the brain has three of these layers called meninges. So let me talk about the three meninges. The first one is just right on the surface of the actual neural tissue, and it's called the pia mater. Actually, maybe let me use a different color so we can stay color coded. Pia mater, it means faithful, and it follows every little nook and cranny of the neural surface. As we go in all, we're going to see these are called the sulci and the gyri. Maybe I should just put that name up here as well. Oops. Sulcus are the little valleys. Gyrus is the little ridge. So it dips down into the sulci and goes up for the gyri. So it basically, and it's just basically this connective tissue, very, very fine layer right on top. You don't even see it. When you are in anatomy and you're looking at a brain and you think you've cleaned the brain totally, taken all of the different membranes off of it, it's still there. It's kind of like, think about kind of spray, a clear coat spray paint on top of the brain. The next layer is the arachnoid layer. Arachnoid, arachnid is a spider. It's because it's got these little web-like struts that anchor to the pia. It does not follow every nook and cranny. It does not follow every sulcus and gyrus. It kind of floats above that all. And there's a space then that gets created underneath it. And that space is going to be the subarachnoid space. That subarachnoid space is actually where the CSF is going to drain into. So there are these little, they're called either the lateral and median apertures, or they're also called the foramina of Magendie and Luschka. But out here in the fourth ventricle, there are these connectors. So the CSF is being produced at the choroid plexus, is inside the brain. But then as it is getting made, you make like a few hundred mils every few hours. So it's overflowing, it overflows into that subarachnoid space. So now the CSF is outside the brain as well as inside the brain. And remember, CSF is a lot like plasma, which is basically kind of like salt water. So what you end up having is the brain is basically kind of floating in salt water. It's got salt water inside and outside, and that's really going to help kind of take the weight off of your brain, right? Your brain is very delicate, kind of squishy tissue. The brains that you played around with in anatomy, those have been fixed with formaldehyde and cross-linking all the proteins and shit. If you have a real brain that's still alive, it's supposedly more kind of like soft tofu. So it's really kind of soft and delicate, but it's floating in salt water. So it, it only weighs like 3% of its actual weight. Think about if you are in a bath filled with Epsom salts or something, and you are just, don't even notice you have a body. You're just kind of floating there. And it's like, oh, wow, it's kind of fun. It's great. And then, I don't know if you've ever done it. You like kick out the plug and just stay there as the water drains. And it's like, at some point you hit the bottom of the tub, and at some point you really start feeling your weight against the bottom of the tub, and it's like, oh man, I forgot I weighed 150 pounds. So your brain is basically floating inside your skull like that, kind of weightless. And so it helps kind of make it weightless, cushions it up in there. In fact, we'll talk, if you end up getting a spinal tap where they take a little sample of the CSF, you have to basically lie flat for a few hours and let the CSF regenerate before you sit up, or you'll get these excruciating headaches and stuff. The next layer, this is arachnoid layer. There's one more layer, which is called the Dura Mater. So what color do I have left? I can do black. Dura Mater. Dura means like tough mother. It's basically dense, irregular connective tissue. It's kind of like leather. It's like a thin leather, and it is the layer that's on the very top here. It's kind of like your brain is in this kind of leathery bag. The one thing that we need to add in about the Dura Mater, though, is it has these little tunnels. So Dura Mater is actually 2 layers inside your skull. There's going to be one layer that is against the inside of your skull bone, one layer that's on the arachnoid. And most of the time, they're just together. You don't even know it's 2 layers. It just looks like one layer. But some places, they separate and you have these tunnels. Like a little tunnel. So a lot of the Dura Mater has these little tunnels that go through, and there is a way for CSF to get from the subarachnoid space into that tunnel. So like if this is my arachnoid layer here, this is my subarachnoid space. It has CSF. There are these little valve-like things called arachnoid villi. So this is going to be another part of the CSF circulation. The CSF is going to, remember I talked about the CSF inside the subarachnoid space kind of floating the brain, but now it's going to drain from that subarachnoid space into these dural sinuses. And these dural sinuses all ultimately drain into your venous circulation. These all drain into the jugular vein. Remember I said CSF is basically like plasma. So if you add the CSF back into your bloodstream, it just basically spit in the ocean, right? It's just adding a little extra plasma to the blood. So if we put this in this other picture here, here's a dural sinus. Let me add those little villi in there. So the basic flow of CSF is going to be like this. One, produced at the choroid plexuses in the brain. As it keeps getting produced continuously, though, it has to go someplace. So 2, it's going to drain out of those little, internal places through these little median and lateral apertures, which are just basically little tunnels, into the subarachnoid space. So 3, it is now in the subarachnoid space. But now it still needs to go someplace. So 4 arachnoid villi drains CSF into the sinus, the dural sinuses. And there's a few of them. There's not just one. There's the superior sagittal sinus, inferior sagittal sinus, the transverse sinus, the occipital sinus. Yeah, again, if you've been in anatomy, you know all the sinuses, all the dural sinuses, but for our class, don't worry about it. And then finally, 5 is going to be sinuses drain back into venous circulation. Right? Because the sinuses basically dump into your jugular vein, which just adds into your regular circulation. So is this making sense? Obviously, you need to have this going. You don't want to block it. In fact, let me. Usually these books have a horrible picture to show you. Oh, they don't. All right, I'm going to share my screen now on my computer. Just to kind of give you a sense here. Right, in this picture, these bluish areas are actually supposed to be the ventricles, and just so you can see, the reality of it is they get very distorted as the brain gets all crazy and bent out of shape. But all that blue in this picture is just representing those hollow spaces. And within there, on the floors of the lateral ventricles, on the roof of the third ventricle, and here in the fourth ventricle, that's where the choroid plexuses are that are making the CSF to start the whole process. Here, this is showing the arachnoid villi as the CSF drains out of the inner places. It ends up in the subarachnoid space in this place, which is drawn in green. And then from there into this dural sinus, which is drawn in blue. And then again, like I mentioned, from the dural sinus just ends up dumping back into your general circulation, into the jugular vein. Yeah, it's just kind of showing the pictures like that. This is showing astrocytes helping make the blood-brain barrier. Astrocyte, again, looks like a little star with all the little processes grabbing onto a capillary. The last thing I'll mention sometimes it's clinically useful to have a sample of CSF, to see, is there blood in there? Is there bacteria? Is there something wrong in the CSF? And obviously you'd rather not have to stick a needle into the center of the brain, like, so there is actually a kind of, it's still invasive, but a lot less invasive than sticking a needle into the center of your brain in order to get a sample of your CSF. And that's the lumbar puncture, or spinal tap. So let me just describe that briefly. Then we'll be ready to take a little break. All right, so here's the brain and the spinal cord. And the spinal cord is going down through your vertebrae. right? If you look at a vertebra, there's like a hole, there's a tunnel where the spinal cord goes down, and then at the bottom is going to be your hip bones and your femur bones. All of these layers I've been talking about, the pia right on there, the arachnoid layer, they cover the whole CNS, including the spinal cord going down the tunnel in between through your vertebrae. The spinal cord itself ends around lumbar 1 or 2. So lumbar vertebrae, you've got like your lumbar, there's 5 lumbar vertebrae and then 5 sacral vertebrae. So your lower back, and then there's the ones that actually make up your sacrum. So the spinal cord ends about here, but then there are these nerves that continue down to go out at other levels lower than lumbar 1 or 2. It's called the cauda equina. If you've been in anatomy, remember the cauda equina. It's just that massive nerves that are running down below the conus medullaris of the spinal cord. But what's nice here is this subarachnoid space is filled with CSF, and it extends down below the very bottom of the spinal cord, which means that you can go in and take a big needle and make it fatter. And as long as you're below lumbar 1 or 2, you're not going to actually be in danger of damaging the spinal cord because you're below the spinal cord and the little nerves are thin and floppy. They'll just get out of the way. So you can actually stick this into the subarachnoid space and suck out a little bit of cerebrospinal fluid for analysis. That is what's called a lumbar puncture. Also, this is a spinal tap. Obviously, you're only going to do this. It's still pretty invasive, but you can go in through the like there's cartilage and connective tissue and stuff that is in between the successive vertebrae. So you can actually just kind of push through and get in there. And like I said, usually people have to kind of stay laying down for a few hours after this and let the CSF build back up in order to have everything kind of full levels again. Again, the cerebrospinal fluid, depends what book you're reading, but it's somewhere like about a half a cup every like 4 or 5 hours or something is getting made. I should show you before we take our little break. The last thing I'll do is we'll look at hydrocephaly. So if you have some blockage of that drainage of the CSF, particularly during development, embryological development, remember the skull bones are not bones when you're still a fetus. They are just membranes. If you remember from anatomy, the intramembranous ossification of your flat bones. So, the brain does have place to expand if you are a little, fetus developing. So the basic idea is these ventricles don't have the drain. So instead of draining properly, they just start inflating. And as they inflate, they are going to create, you can see these pictures here. Like just the whole head ends up becoming like a balloon. That's horrible. I don't even want to look at that. But what they can do is they can go in and they can add in these shunts and create drainage. So, things can then work normally or drain normally. So proper drainage. Yeah, some of these pictures are pretty disturbing. So let's stop sharing that. But just to give you a sense that if that drainage is not working properly, it's not good. You can actually have your brain is pretty darn plastic in the very beginning. So if things are caught really early, the brain is still undergoing a lot of development and things can proceed pretty normally. Like if you have damage to an adult brain, you're in much worse shape because you have much less what we call plasticity, the ability to kind of rewire and reconfigure things. But at the very beginning, your brain it's got like twice as many neurons as you're going to need as a adult. So there's still a lot of pruning and rewiring going on.